You've pushed the envelope in clinical trials in the UK with a 4-drug regimen. Tell us about that, Gareth, if you don't mind.
So we introduced carfilzomib, Revlimid, cyclophosphamide, and Dex as one of the major induction arms, and I think there are about 1,000 patients in that, and it was well tolerated, no cardiotoxicity, excellent responses, and everybody was able to go on from that to the transplant. So I think there is some merit in 4-drug regimens, especially in younger, fitter patients.
Panabinostat, you were telling me earlier that you use this in your 3rd line.
Yeah. I mean, I think, you know, the challenge with panabinostat is that there is some GI toxicity. So you have to figure out the right schedule, and then you have to figure out the right partner. And I'll be honest, I don't think bortezomib is the right partner. I think there's just too much overlapping GI tox. So we've been able to use carfilzomib as the partner, and then with the 1 week on, 1 week off, 1 week on schedule of panabinostat.
So what dose of panabinostat?
20, 3 times a week.
Anybody else using
And that's a
panabinostat?
per day hospital.
Yeah. So I've utilized, you know, the K, panabinostat, dexamethasone in our, you know, 3rd or 4th line relapses, and I agree. You know, the Monday, Wednesday, Friday, 20 milligrams schedule along with K appears to be well tolerated, and it's got reasonable efficacy too.
What about second transplant, Ivan? Are you still in favor or against those if people have done well with the first transplant?
So, I mean, we have transplanted people a second time, but in general, it would be people that have achieved at least 5 years or more from their first transplant are the people that we would recommend a second transplant to.
Same all over, people doing second transplants?
It's
Tom?
pretty interesting. So we collect enough stem cells up front for 2 transplants, essentially for everybody. And if we look at our database and say how many really got a second transplant, it's less than 5% of patients.
Yeah, we looked at that, about 10%. It was shocking, really. You'd think it would be much higher.
Right.
Why is that? Because we have better drugs, or we're just not doing it? What's the...
I do think that we have better drugs that are less toxic than a second transplant. You know, it's a last-ditch effort in my mind. Patients that have had all these drugs and now have cytopenias, and you have some stem cells sitting in the bank, I think it's a great time to give them a melphalan-based therapy and then give them back them stem cells so they can get their counts back up and they'd be eligible for clinical trials again, and et cetera.
And that's our most common indication for
Agreed.
second.
I think that is probably the most common salvage indication. And you're not going full dose mel with those patients.
So
you know,
I'm
you're
like
just
a bit of a kind of outlier yet again, it would seem, in that I think there's quite a lot of efficacy to be had for transplant at relapse, especially if you haven't seen therapy for a while or you've been on non-alkylating agents. You can adjust the dose, support the tox using stem cells that are stored at presentation. I think it offers a way of shaking up the clonal composition of the tumor, and then as long as you have some form of maintenance strategy afterwards, I think it's more than a reasonable approach.
But what's your time cutoff? How long?
So we were using an 18-month cutoff in the UK. So if you've got 18 months, I produced a set of data that's published that says they benefit from a second transplant compared to people who didn't. I think 5 years is a bit long, but times have changed, and if you're on maintenance for 2 or 3 years, 5 years maybe you know, from presentation.
So maybe we can compromise at 3 years. How about that?
Yeah, 18 months. I mean, 18 months, to be honest and fair to your study, they didn't have access to all the stuff that they often do.
It was historical.
Yeah, it's hard to extrapolate.
We still use second transplants. I was actually surprised it was only 10%. It feels like more than that. Talk about sequencing. We started off talking about induction therapy, and we said that, I mean, do you think monoclonal antibodies are going to move into the frontline setting? I guess that's the first question.
Yeah, I mean, certainly the data in the relapse setting looks very promising. Certainly with daratumumab it does, and I think there are early studies being presented here at ASCO this weekend looking at DARA combinations that we've discussed earlier, still in the phase 1 setting, but I think it's only a matter of time before that data matures and then we do the sort of definitive studies in the upfront setting.
And what's going to happen with sequencing of therapy when that takes place, this transition to, you know, 2 or 3 drugs plus an antibody in the newly diagnosed setting? Do you think we'll be retreating with monoclonals, or are we going to be switching to newer drugs? What do you think?
I think we'll have another panel discussion asking the same question. I think it's going to, no, I mean, seriously, I think it's going to be driven in large part by the data. And I think it's going to be driven by what new agents are on the market. And I think it's also going to be driven by biology. I mean, I think the one thing that we're learning more and more, I mean, like with the Arkansas molecular profiling, is that myeloma is not one disease, and there may be some patients that are more likely to respond to proteasomes, others to monoclonal antibodies, others to different forms of immunotherapy, and I think we are going to have more of a precision medicine guided by biology as opposed to empiricism that we're currently doing now.
It's very hard to be prescriptive these days when you're asked, how do you treat relapse? Because it's almost unique to every single patient.
Absolutely.
Would you agree, Saad?
I agree with you. I think that there are so many factors involved, disease biology to begin with, and the disease biology that you're dealing with at the time of relapse, how patients have been treated, what kind of tox they had, and all of those go into the, factoring in what therapy to give them.
Any other thoughts on treating relapse that we haven't covered?
I think I'd like to develop that point that Saad just hit on about, so it's at some level people don't think about molecularly reassessing patients at relapse, and I think we have to. We have to know if they have p53 mutations, what molecular subtype they are, the nature of the relapse, and it is becoming a field where we tacitly use precision medicine or personalized medicine, whichever way you wish to describe it.